Hello, everyone. My name is Callie Nest. I'm a student in your biological diversity class, and I'm going to be talking about chapters 26, which was about bacteria and archaea, chapter 27 with eukaryotes, more specifically all about protists, and then a little bit of what we covered in class about chapter 28, which is about plants. So this is just going to be a review of mainly what we covered in class and then supplemented by information from the textbook. So for chapter 26, like I said, that was bacteria and archaea. They're both prokaryotes. They both lack a membrane-bound nucleus, and they make up our microbiome, which is just a community of microbes in an area. So microbiomes in your gut, microbiomes on your skin, in your mouth, and then microbiomes all across the world. And then if we want to break down some different molecular features, both bacteria and archaea have one type of RNA polymerase. Bacteria has 5 subunits and archaea has 13 subunits, which is similar to eukaryotic RNA polymerase 2. Bacteria has peptidoglycan present in the cell wall, which we will get to a little bit later when we talk about gram staining, and archaea does not have peptidoglycan in the cell wall. The first amino acid incorporated during translation for bacteria, it's formylmethionine. For archaea, it's methionine. And then are histones associated with DNA? Bacteria, no. Archaea, yes. And why are they important to study? They're abundant. They're absolutely everywhere, inside the human body, on the human body, in animals. They have a relationship with every other kind of organism. We talked about extremophiles, which are organisms that live in extreme conditions, such as areas with low pH, hydrothermal vents, extremely cold or hot temperatures, and salty water. From studying extremophiles, we were able to find a heat-resistant enzyme for polymerase chain reaction, and then studying extremophiles supports our search for extraterrestrial life, as well as understanding the origin of life on Earth. Of course, bacteria and archaea are medically significant. They can be pathogens. We talked about antibiotic resistance, we talked about biofilms, and we talked about endospores. When we talk about pathogens, we want to talk about virulence, which is a heritable trait that can lead a specific strain of a bacteria to be a pathogen. We talked about endospores, which are structures created during periods of environmental stress. So these are just containers with copies of the cell's DNA, RNA, ribosomes, enzymes. Endospores are resistant to high temperatures, ultraviolet radiation, and antibiotics. And then if environmental conditions become favorable, they can actually start to reactivate, and cell division can begin again. We talked about biofilms, which are colonies of bacteria in a fatty matrix, which protects that community from antibiotics. And it excretes extracellular polymeric substance, which is that sticky stuff that we talked about. Why this is important is that this can be found in hospitals, and it's extremely hard to get rid of. And then an environmental impact of bacteria and archaea is bioremediation efforts, which deals with the ability of microorganisms to break down pollutants. And to support bioremediation efforts, we talked about biostimulation, which is adding nutrients to a polluted area where there are already microbes that are working on breaking down those pollutants. And then seeding is when we introduce those microbes into areas that are polluted in the hopes that they will be able to break down those pollutants. So how do biologists study these organisms? We talked about enrichment cultures. That's where you establish a series of environmental conditions to keep stable, to study how cells grow and react to those conditions. We talked about metagenomics and environmental sequencing, which is where you catalog all the genes present in a community. And then, as I mentioned before, we talked about studying microbiomes. One method of discussing diversification is by understanding gene transfer. We talked about 3 kinds of gene transfer. First one is transformation. That's where a bacteria or an archaea cell will take up DNA from its environment. So maybe a cell has died, then its DNA kind of floods out, and then another cell can directly take that up. Transduction is where a virus transfers DNA from one cell to another. And then finally, we talked about conjugation, which is direct cell-to-cell transfer of DNA. Bacteria and archaea can be diverse morphologically. They can vary in size and shape and arrangement. We talked about this, which is just different shapes and arrangements of cells. They can have different ways they move about, their motility, flagella, gliding, or they are non-motile, which means they don't move. There's different ways that they can acquire energy. So we broke it down by how they got their building blocks, their sources of carbon. So they can be autotrophs, where they make their own food. They can be heterotrophs, where they have to get food from other organisms in the environment. They can also be mixotrophs, which is a combination of the two. Then there's different ways they can source their energy to make ATP. Phototrophs use light energy. Chemoorganotrophs use organic molecules. Chemolithotrophs use inorganic molecules. So the book gives a lot of examples of specific organisms that are different combinations of these. And it's interesting to look at because there's a ton of different varieties. One way to differentiate different kinds of bacteria is to do a gram stain. And when you do the gram stain, gram positive bacteria look purple, which means that their cell wall has extensive peptidoglycan. Gram negative bacteria look pink, and that means that the cell wall has a thin peptidoglycan layer and an outer phospholipid bilayer. Next, we talked about chapter 27, which primarily deals with protists. Protists are all eukaryotes except for land plants, fungi, and animals. They tend to live near or in water. They can be unicellular or multicellular. We talked about a wide variety of outer coverings for those protists. And they have a lot of importance medically. They can cause diseases, both in people, animals, and in agriculture. They're significant ecologically. We talked about eutrophication, harmful algae blooms, and red tides. In addition, they are primary producers. They play a key role in the food chain, and they can create carbon sinks. And they can also help us understand evolution because they are eukaryotes. So another key topic that we talked about is endosymbiosis. Endosymbiosis is when one organism lives inside another organism of a different species. Why is this important? We talked about endosymbiosis theory, which just says that a bacteria cell once entered another cell and lived there. And this can explain why eukaryotic cells have specific organelles, as well as the behavior of those organelles, such as mitochondria. So some evidence that endosymbiosis may have taken place in what was mitochondria and then a bacteria cell, or inside a protist or eukaryotic cell, is that mitochondria are similar in size to alpha proteobacteria. They replicate by fission like bacteria. They have ribosomes that are similar to bacterial ribosomes. They have their own DNA called mitochondrial DNA, and they have a double membrane. So endosymbiosis theory suggests that mitochondria came from a bacteria cell which entered another cell. And similarly, we can talk about another theory of endosymbiosis with chloroplasts. The theory for that is that a protist ingested a cyanobacterium. The evidence for this, a lot of these are similar as with mitochondria. When we talk about a bacteria cell entering another cell and making its home there. Chloroplasts have circular DNA similar to the shape of cyanobacteria's DNA, and they also share the same photosystems as cyanobacteria. And then another thing mentioned in the book was that chloroplasts with their multiple membranes may have been acquired by secondary symbiosis, which basically means endosymbiosis occurred twice. So you had one organism entering another cell. So then you have that, let's call it a huge home cell. Then that cell was ingested, or it entered another cell. So you have a cell within a cell within a cell. Another origin theory that we talked about was the nuclear envelope and the endoplasmic reticulum. We discussed that this may have been from an infolding of the plasma membrane. This is important because this separates the locations of transcription and translation. And then we talked a lot about innovations in the ways that protists protect themselves, like with their outer coverings. I'm going to talk about the ways that they move, how they get food. Some of them are multicellular, and then also they're diverse with their reproduction. Three main ways we talked about to get food is by ingesting. So this is when the protists eat live or dead organisms, or they scavenge. So they can have a wide variety of mechanisms in which they can ingest organisms. They can absorb nutrients across their plasma membrane from their environment, and of course, they can use photosynthesis. Protists are diverse in their movement as well. We talked about amoeboid motion, which is sliding using pseudopodia. They can have one or more flagella, many cilia. They can be sessile, which means that they don't move, or they can be planktonic, which means that they're just floating in water or the ocean. So two ways that they can reproduce is either by asexual reproduction or sexual, wow, I can't say that word, sexual reproduction. Asexual reproduction is via mitosis, and mitosis produces genetically identical daughter cells. Sexual reproduction is through meiosis and gamete fusion, which produces genetically different daughter cells, which is where things kind of get a little complicated because we start to talk about life cycles, and life cycles are extremely important going forward. We're going to talk a lot about them with when we go over plants and then continue on with other kinds of organisms. The way I like to look at life cycles is to start from a mature cell, but you can also start from a zygote. This picture I just drew the mature cell. So we can see that the mature cell is haploid, it's N. If we go to the right, it divides by mitosis, so it's creating genetically identical daughter cells, which are also haploid. These will grow and mature again into mature cells, also haploid. On the left, so down here we have the mature cell going through mitosis. It produces gametes, which are haploid. Then we have fertilization. So we have two gametes combining. The gamete pair fuses and it produces a diploid zygote, which through meiosis produces haploid cells, and these cells grow to mature. Now we can talk about diploid dominated life cycles. Again, starting off with a mature cell, this time it's diploid 2N. It goes through mitosis for asexual reproduction. So it will continue going through this loop of mitosis and maturation until the cells are too small to continue dividing. Then they will go through sexual reproduction. So they will go through meiosis to produce haploid gametes. Two gametes will fuse and you will have fertilization, which produces a diploid zygote. This zygote grows and matures into the mature cell, and then we can go over the process again and again. Continuing on with life cycles, which as I talked about is really important with plants, just some terminology. Alternation of generations is when one phase of an organism's life cycle is based on haploid form and another is based on diploid form. Other words that you need to understand, gametophytes, it's a multicellular haploid form. It produces gametes by mitosis and cell division. Then gametes fuse to produce diploid zygotes. Sporophytes are multicellular diploid forms. They produce haploid cells or spores via meiosis, and then spores are single haploid cells that divide via mitosis to form haploid gametophyte. So I find life cycles to be a little confusing. Just because there can be a lot of terminology introduced into it. But the key thing to understand is how to go between gametophyte and sporophyte forms. And you can follow this with the changes between haploid and diploid forms, and then also following the patterns of mitosis versus meiosis. So here is a visual guide to alternation of generations. Starting at the top, we have the gametophyte. It's haploid and it goes through mitosis to produce gametes, also haploid. So then we will create a sporophyte, which is diploid from these gametes. The sporophyte will undergo meiosis to produce haploid spores. So this is essentially how the different forms can alternate. So like I said, alternation of generations, one phase of life cycle is based on haploid form, another is based on diploid form, and we can see this as we go from haploid to diploid. Plants, we're talking specifically green algae and land plants. That's how the book divided it. Similarities between green algae and land plants. Their chloroplasts contain chlorophyll A and B and beta carotene. Their thylakoid arrangements are similar. Thylakoids are membrane-bound compartments in chloroplasts that form stacks called grana. They kind of look like pancake stacks, and understanding the different parts of a chloroplast are important for understanding the mechanisms behind photosynthesis. Other similarities between green algae and land plants. So their cell walls are similar, the sperm is similar, and peroxisomes are similar. And going back, way back to the beginning of the semester, it might have been like chapter 1, when we talked about cells, peroxisomes oxidize different chemicals in a cell. Finally, chloroplasts synthesize starch for energy storage. And then why is it important to learn about plants? I mean, there's so much. I mean, pretty much everything in life is bound to the function of plants in some way or the other, whether you eat them or whether the food that you eat eats plants. I mean, you need oxygen to live. They support climates. They hold soil and water. They're extremely important in carbon sequestration. And then we can make a lot of important building materials out of plants, and also medicines. So finally, we started talking about the difference between vascular and non-vascular plants. Vascular plants have vascular tissue, which are specialized cell groups that transport water and nutrients from one area of the plant to another. Non-vascular plants, in contrast, do not have vascular tissue. So they are limited in where they can live and limited in their shape, because they need to be able to function in such a way that water can adequately travel to all of their cells. And water has to basically be transported against the force of gravity. If gravity is going down, then water has to go up and through the tissue of plants. So there's different ways that plants can do this. Seeds contain a plant embryo and nutritive tissue, all surrounded by a protective layer. And then we can break down the two types of vascular plants that use seeds. Angiosperms are flowering plants, and gymnosperms are plants that produce seeds and cones for the most part. Seedless vascular plants such as ferns rely on spores, which, as we talked about earlier are single haploid cells. And then non-vascular plants don't use seeds, they use spores for reproduction. Okay, so this is essentially where we stopped talking about plants for this week in lecture. Next, it's really important to understand the transition to land and how evolution played a role in plant form and function. One important characteristic in living on land is preventing water loss. So plants have a cuticle, which is a waxy layer preventing water loss from stems and leaves. And then we talked about stoma, which is an opening surrounded by two guard cells that can open and close to control gas exchange in photosynthetic tissues. So that's it. Let me know if you have any questions, and I really appreciate feedback, and I hope you have a great day.